record. Okay, so today we are starting a brand new kingdom, and that's the kingdom which we belong to. It's the animals, and then there will be two very large groups now known as the Protostomes and the Deuterostomes. It will be on the lab, on the lecture exam and the lab practical. So you need to understand what they are. I'll give you the definitions, and then we'll talk about the presence or absence of body cavity. Well, if there is no body cavity in the organism, we're talking about acoelomates. Xylem is nothing else but the body cavity. So you're saying body cavity means xylem. You're saying xylem means body cavity. And I'll talk about the different types of the body cavities. There is three: no, false body cavity, and the true body cavity. And based on that, we have several groups of animals broken down based on that, as well as the embryonic development, which gives us protostomes and deuterostomes. Both of those topics will be on the lecture exams, on the lecture exam number 3, as well as the lab practical number 3. So this is basically what we are looking at, something which is known as the animal kingdom. And animal kingdom will be broken down into several major groups. Well, first of all, remember we have that particular branch. Fungi come out, and then we talk about the choanoflagellates. Choanus means color, and they have flagella, so that's a very interesting protist, and genetically they're fairly close to animals. Then we have a group known as the sponges. Sponges are not true animals per se, because one of the characteristics of animals are true tissues, a specialized group of cells performing a particular function. Peripheral doesn't really have those. They do have some of the cells, and those particular cells which perform a particular function, let's say they have choanoflagellates. And those particular choanoflagellates at some point of time can change into a different time for the different type of the cells in the periphery. And we'll discuss that briefly. And then the rest of them known as something which is known as the metazoa, or true animals. And there we have a group which is generally radial symmetry, and most of them are bilateral. Here's a tiny little bit of problem with the bilateral. In bilateral, we will have a really interesting group known as the sea stars. And the sea stars, or echinoderms, sea stars, the sea urchins, etc., all of the relatives. So they are actually radial symmetrical as the adult, but they are bilateral symmetrical as the larvae. And we'll talk a little bit about that later on, probably next week. So, and then of course, we will have several major groups based on the presence or absence of the xylem. And then, of course, we are looking at the deuterostomia and the protostomes, and we'll discuss that in length. So, in order to belong to the kingdom of animalia, to the kingdom of animals, beasts, all of them are eukaryotic organisms, means that they have, well, cells have a nuclei, usually a nucleus, and membrane-bound organelles, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, etc. So all of those particular complex organelles. Now, they are multicellular. So remember, if it's look like an animal, but it's a single cell, it's automatically protist. All of the animals have more than one cell. In some cases, it's literally like 100 or so cells. In some cases, look at the human being, 100 trillions of cells, but still a multicellular organism. Now, heterotrophic, we have to consume something in order to get organic compound. We cannot just go outside, bathe in the sun, and get that particular sugars made right in us. No, we cannot. We have to consume pre-made organic compounds. And who are making them? Oh, predominantly plants and bacteria. This is the two major groups. Well, some of the bacteria, not all of the bacteria. Some of the bacteria, photosynthetic bacteria. So those groups known as the producers, and we, as a part of animal kingdom, are consumers from the ecology point of view. Now, remember we talked about specialized cells, cells which will perform a very specific function. They will specialize. Muscle cell versus nerve cell. Very different looking cells, very different functions, yet they have pretty much exactly the same, not pretty much, exactly the same DNA. My muscle cells will have exactly the same DNA as my nervous cells, as my skin cells. So, but they are specialized to perform a particular function, all right? And they are, like muscle cell at some point of time will not be able to convert into a, let's say, blood cell, vice versa. Blood cell will not be able to convert into a nervous cell, so anything like that. Next thing, capable of locomotion, meaning they can move at some point of their development. The larvae or the adult, we can move as the adults. In some cases, let's say corals. Corals don't really move much as the adults, but the larvae can swim fairly freely. So in this particular case, locomotion, movement, this is how we interacting with the environment. Remember, plants are growing into the environment. They do not move. Well, they move a little bit, but they will not be able to cover, let's say, the body length of the plant within a couple of minutes. No, that's not happening. We, however, as the animals, can do that. And this is how we're interacting. We are moving into the environment instead of growing into the environment. Can respond adaptively to the external stimuli. External stimuli is basically any kind external irritant which produce some sort of response. Any kind of changes in the external environment which are producing response. You smell something bad, you're like, hey, here we go. By the way, remember the ginkgo plants? Yes, females, yeah. Seeds, yeah. Will be on the test. I think it's on the test. So it's definitely on face-to-face lab practical. Yesterday I was setting that one up. Anyway, so we are responding adaptively. I'm telling you something, you are following the instructions, hopefully. That's the response to the stimuli. Can reproduce sexually. Most of the animals will reproduce sexually at some point of time. If there is an asexual reproduction, yes, there is an asexual reproduction for animals, but most of them reproduce sexually at some point of their life. Do animals have alternation of generations? Nope. Not at all. We are completely different from that particular point from the plants. Plants have alternative of generations. Gametophyte will produce sporophyte. Sporophyte will produce gametophyte. Diploid stage will produce haploid stage. Haploid stage will produce diploid stage. We don't have that. As a matter of fact, the grown organism will produce sperm and egg through the meiosis. So now typical regular meiosis in animals. So sperm, egg, or both, there is a hermaphrodites. And then whenever the sperm and egg haploid and haploid structures will form, will unite, the sperm will fertilize the egg. It will form a zygote. Zygote is a single cell at that particular point, single cell organism, but it's a diploid. All of us started as the zygote, about 30 or so minutes of first 30 minutes of my life, your life, we spend as a single cell organism. And it's important because basically we are, during the embryonic development, we kind of repeating the steps of our ancestors, long time ancestors. We start with the multiple cells, then we start dividing, with a single cell, then we start dividing. We ended up in a colony of the multiple cells and so on and so forth. And we'll talk very briefly about the embryology. As a matter of fact, the first lab, which we'll do next time, is has a part of the embryology. This is where we'll talk about the blastula, gastrula, there is a morula, and we'll discuss that a little bit in a greater detail, and what exactly, which particular layers will give us which particular systems. Very important. So in this particular case, that statement says multiple cell division results in a blastula. So remember, diploid zygote. Now we'll start dividing as a single cell, but 30 minutes later will become 2 cells, 4 cells, 8, 16, 32, 64, 128, 256, 512, 1024, and so on and so forth. So it's not really growing per se. The size, yes, it grows a little bit, but the cells continue to divide. So you see that particular very large cell. Now you have 8 cells, which are much smaller. So, and in this particular case, we're talking about mitosis, cleavage, really, really fast. So at some point of time, it will create the ball of cells, that particular ball of cells known as the morula. As the cells continue to divide, continue to become smaller and smaller and smaller, that particular ball of cells will turn into a hollow ball of cells. There will be a cavity inside. And that particular cavity inside, known as the, well, that particular stage known as a blastula, when we're looking at the hollow ball of cells, okay? And this cavity will be known as a blastocell. So now remember, xylem is a cavity or body cavity. So in this particular case, blastula cavity within the blastula known as the blastocell. Now, we have a hollow ball of cells which continue to divide. The cells are continue to divide. As a result, the walls of that particular blastula at some point of time will start collapsing, moving in. And that process, whenever the cells start kind of collapsing and moving inside of the blastocell, known as the gastrula. And then it will continue to collapse until it will reach that particular, the opposite end, so to speak. And that tube, which is kind of collapsing, went through the whole ball of cells known as the archeron, which is the ancient gut. That part will develop into a digestive system. All right, but I'm kind of getting ahead a little bit of myself. So in this particular case, what you will need to understand that we have a diploid organism or diploid structure zygote, which continue to divide. Then at some point of time, it will turn into a hollow ball of cells. That's important. Actually, I will tie it up to the evolutionary development of the animal. So that particular blastula continue to divide, and then at some point of time, some of the cells will start moving inside. And that particular movement known as a process of gastrulation. And that particular opening will either, remember, the whole tube will create a digestive system. Digestive system have a beginning and have an end, have a mouth and have an anus. So 2 groups of animals, which I already kind of addressed to you, protostomes and deuterostomes, will be subdivided based on what this particular initial opening will turn into. Protos from Greek, I think it's a Greek name, a Greek word, protos. Protos means first or initial, okay? Stomos means mouth, stomatology, right? So in this particular, same root. So in this particular case, if that initial opening will turn into a mouth and then the digestive system continue to grow, and then whenever it reaches the opposite end, that particular opening will become anus. That's a protostome. Now, the other group of animals decide, well, they didn't really decide, but they are like, okay, that particular initial opening will become an anus. And then the continuation, the secondary opening will turn into a mouth. Deuterous, secondary mouth. Mouth will develop second, all right? So here we're talking about the protostomes and deuterostomes. Difference in the cleavage, etc., and we'll discuss it at length. Not too much, but it will be also on the lab practical and lecture exam number 3. All right, so here we have all of the eukaryotic groups which we kind of covered. Most of them, well, some of the genes are shared among all of them. But, as you can see that fungi and the metazoa are sharing more genes, the overlap of those particular circles or ovals between the fungi and the animals, true animals, are much larger than, for example, animals and the plants. So in this end, that we're calling it, this is basically the protists. So as you can see, we have a very distinct set of genes of our own, but some of those genes we're sharing with all of the eukaryotes, and some of them, or many of them, we're sharing with the fungi. So that's generally how that particular tree of life looks like. So this is 4 major kingdoms, which, well, 3 of them we are covered. We covered the protists, we talked about the fungi, we talked about the plants, and now we're talking about the animals. That's another representation of the same thing, just in the tree format. As you can see, common ancestor at some point of time, common ancestor give us branch, and that particular point of time we're talking about 2 or so billions of years ago, probably. So give us the branch which turned about 500 millions of years ago into a green plant. Remember Cambrian explosion. Then there was a, well, that was a split, archaeoplastids, it's still protists, right here at that particular point. And then we are ended up into a branch where we and the fungi located that. And that split happens a billion, maybe 2 billions of years ago, 1.5 billion, probably. So animals have a diploid life cycle. So whenever you're looking at the animals, unless you're looking at the egg or the sperm, you're looking at the diploid organism. Easy comparatively to the plants, right? Only haploid cells are the gametes, means egg and a cell, egg and a sperm. The male provides the sperm, which is fertilizing the female's egg. Remember, some of the organisms are hermaphroditic, meaning they have, at the same time, they have the male and female gonads. And later on, especially the simpler organism, later on we'll talk about the some of the parasite worms. Most of those parasite worms are hermaphrodites. Not all, but most of them. So fertilized egg called a zygote, and that's already a diploid. So everything is good, which develops into a multicellular embryo and eventually become a new diploid organism, which will grow into the adult, and then it start producing gametes, one or another way, or both. So far, so good. Any questions, any problems? So once again, no alternation of generations. On Friday, I was hosting, well, I was in bio lab for a lab practical review for the face-to-face classes, and I had several students asking me, so for the plants, there is a gametophyte, sporophyte, who am I? I'm like, well, you tell me, who are you? Are you a gametophyte or sporophyte? And the lady like, well, I'm producing gametes. I'm like, yes. So I am a gametophyte. And I'm like, yes, technically you're right. You are producing gametes, so you have to be a gametophyte. But on another hand, you are diploid. All of the plants, all of the plants, gametophyte plants are haploids. So what's wrong? And she's like, so I'm sporophyte. I'm like, well, no. She's like, what does it mean? I mean, phytos means plant. You're not plant. So you cannot apply the characteristics of one kingdom to a totally different kingdom. We don't have that alternation of generations. So keep that in mind. All right, so origins. Origins are quite interesting, and we are talking about at least 700 million of years ago, probably closer to 1 billion of years ago. But again, if you'll remember, or you might not know, about 1 billion of years ago, we had a very interesting thing. One of the very interesting things was happening was global cooling, ice age, a global ice age. And we're not talking about too many glaciers. The whole Earth was covered by the ice, something which is known as a snowball Earth. And roughly 700 to 600 millions of years ago, one of those particular stages of snowball Earth start melting out. The temperatures on the surface warmed up, and we start seeing what? We start seeing the explosion of life, Cambrian explosion. Speaking of which, there is a short, I send the links to the short videos. They are about 10-15 minutes each video. And during this particular unit, I would ask you to watch those videos. I'll see how the first couple of them goes. And if I'll see that there will be a low participation, I would have to, and I already have those, I would have to go with the homework, well, with the worksheet assigned to those particular videos. Do you really want an extra paper to fill it out and submit it, and then you'll submit it in a Word format? I'll give you a 0, you'll become all upset. There is technically no reason for that. I'm asking you just to watch it. It will help you tremendously to understand that particular unit. And again, videos are short and quite interesting from scientific point of view. They're not super boring. I will guarantee you that. I specifically chose those videos. But once again, please watch them. It will talk about the animal genes. It will talk about the Cambrian explosion. So they're quite interesting from the point of view of science. And once again, they will give you a fresh perspective on what animals are. So please do that. I really don't want to create extra work for me and for you, because extra worksheet, you'll have to do it, then I'll have to grade it. So all that stuff. Please watch. But in this particular case, we start talking about the choanoflagellates. We already discussed the choanoflagellates. I'll show you the picture of the choanoflagellate. And there was a evolutionist, the gentleman known as Ernest Haeckel. He was a zoologist. Actually, he wrote a very interesting zoology textbook. So what he proposed is that we had a colony of the choanoflagellates, kind of hollow ball. Remember, we already heard about today about the hollow ball, the blastula. So this is based on the blastula. Ernst Haeckel says, well, at some point of time, probably it was the free-living colonial organism, similar to what we saw in Volvox, but Volvox was more like a plant, a green algae. But in this case, we're talking about the hollow ball of cells, which was kind of floating around. Of course, that's in somewhere in aquatic environment, floating around. And at some point of time, the bottom, since it was kind of gliding along the substrate, some of those particular cells on the bottom of that particular ball become more specialized. They were grabbing and digesting. As a result, we start seeing those cells are moving inside. Now we're looking at the gastrulation. So this is what was proposed. Again, choanoflagellate, the cell, nucleus, that particular color, very interesting structure, and the flagellum. Here we're looking at the microphotograph of the free-living choanoflagellates. Again, cell, color, and the flagellum. So what basically Ernst Haeckel proposed, that the group of those cells later on start specializing and we start seeing some of the specialized cells for digestion start moving inside of that cavity of the hollow ball. So we start looking at the first specialization, the movement and protection for the outer layer, and then the digestion, and then distribution of the nutrients for the inner part. We start looking at the first two embryonic layers. So here's what we are looking at. This is where the choanoflagellates, as the protist coming out, as you can see, we're sharing even more similar genes comparatively to the fungi, and definitely much more comparatively to the plants. So they are on the same kind of branch of that evolutionary tree. Comparative genomics is very, very similar to what we see in animals, the ancient genes. So here's what we are thinking was happening. Choanoflagellates, a hollow ball, again, representative by the blastula in the early embryo stages of modern animals. And some of the cells become more specialized. Why? Because they are closer to the surface. This is where all of the organic compounds or little bacteria, little algae can be found. So it will be grabbing those and then they will start digesting. So they had predominantly digesting function. So in this case, those particular feeding cells, and of course they will spread that particular extra nutrient to the other cells, sharing, calling, right? So in this particular case, we start looking that those digestive cells start moving inside. Easier to share once again. And now we start looking at the digestive cavity. It's not a body cavity, it is the digestive cavity. So those cells are moving, the whole organism, and some of the particles, food particles getting in, and those cells are producing the digestive enzyme, digesting whatever is ended up in the digestive cavity, sharing the nutrients with the rest of the body. Now look at this particular creature. Now add a little bit of tentacles, and you are looking at the jellyfish body plan, something which is called medusa body plan. This is how the first group of the animals, true animals, look like. 2 layers, and then the kind of digestive cavity, a sac of the digestive cavity. Whatever is coming in from the mouth, then whatever is not digested will come out from that particular opening as well. Not a complete digestive system. Complete digestive system have the opening where the food comes in and the opening where the food comes out. All right. So keeping that particular thing in mind, now several innovations in animal evolution. Tissue structures, remember, specialized cells. Cells which are specializing in protection, in digestion, later on in the contraction, so the movement will be much more active. Body symmetry. We are bilateral creatures. Well, by we, we're talking about humans. But there is a group of animals which are having the radial symmetry. We'll talk about that today. Presence or absence of body cavities. I already told you there is 3 major groups. No body cavity, false body cavity, and then true body cavity. And I'll discuss that in greater details, and I'll give you the examples. And then later on, I will ask it on the lecture test, short answer right there. Development patterns, how they develop, how the cleavage is happening, and what exactly is the origin of the mouth. Is it a secondary or is it a primary? So protostomes and deuterostomes. And then the presence or absence of something which is known as the segmentation. Segmentations are the repeating unit within the body of an animal. Some of the animals don't have segmentation. We have segmentation. You can say, I don't have segments. Yes, you do. Think about this. Well, let's go with the spinal cord. Spinal cord goes, and then there will be a pairs of nerves coming out from the spinal cord, segmentation. Same thing with the ribs, right? That's the our bones are repeating unit, connecting to each other, protecting our upper body cavity, or whatever the organs within our upper body cavity, right? The vertebral column. repeating units over and over and over again. Yes, they are slightly different from each other, well, in humans, but if you look at the snake, they will be very, very similar to each other, almost identical. So segmentation is important, and I will talk about the segmentation a little bit later. So now, asymmetrical. Please remember that most of the sponges, and for simplicity's sake, we'll say all of the sponges, don't have the symmetry. They grow along with whatever we have or whatever they have in the environment. If the stream goes one way, the sponge goes into that particular. So they don't have a symmetry per se. They might start looking kind of like a radial symmetry, but generally they are asymmetrical. So please memorize, highlight that. The sponge is considered to be non-symmetrical. Asymmetrical, there will be a question on a lab practical, I think almost certain of that. Lab practical 3, not lab practical 2. So radial symmetry. Radial symmetry are phylum Cnidarians, the jellyfish, corals, and their relatives. Sea anemones will have a biradial, still a radial. It's a special type of the radial. And then there is something which is known as the comb jellies, ctenophores. We'll discuss them in the lab. Next lab, yes, lab number 10. And then most of the animals are bilateral. The right side, left side, and the right side are more or less identical. Why I'm saying more or less identical? It's not exactly the same, but very close to that. And again, if you'll start digging deeper, like you split a person in half, don't do that. But for example, the left side of the lungs are smaller than the right side of the lungs. The heart located mostly on the left side of the body, well, of the chest, right? On the right side of the abdominal part, you have the liver. On the left side, you have something which is known as the pancreas. Even though we have 2 kidneys on either side, left and right, one of them is a little bit shifted up and the other one is shifted down. So all of those particular things will not make it as symmetrical per se, but close enough. We're not looking for perfect symmetry, but I have a right hand and left hand, and one eye and the second eye, ears and so on. So more or less the same. Not exactly, not precisely, but more or less the same. So sponges are considered to be asymmetrical, meaning there is no internal symmetry whatsoever. Most of the animals, however, besides the sponges, are symmetrical, and there is 2 major parts: radial symmetry and bilateral symmetry. Radial symmetry, the parts are arranged around the central axis. Bilateral, you have just one central axis, and the left and the right side are about the same, mirror images. So here we're looking at the radial symmetry, right? The central axis, and then you have several units arranged about that. In this particular case, we have 1, 2, 3, 4, 5, 6, 7, 8 ray symmetry. I told you already about the echinoderm starfish. They usually have a 5 ray symmetry, and so on. So bilateral symmetrical shows something which is known as a cephalization. Cephalization is formation of the head. Front end, when goes into the environment, it have to sense the environment. If there is a danger, if there is a food or anything like that. So sensing of the environment, how it's happening. There will be quite a few different sensors on the front end of the body, all right? We're talking about temperature sensors, smell sensors, light sensors, well, chemical sensors, it's a taste and a smell at the same time, essentially, and so on. Pressure sensors, pain sensors, etc. So those particular sensors will be predominantly found in the front end. Front end, now, getting all that information from the environment, because this is the first part which goes into the environment. You have to sense it. Now, whenever you're getting all that information, it have to be processed. And as soon as it will have to be processed, we start talking about formation of some sort of brain. Don't have to be a complex brain, but still, all of this information need to be processed. So the organism will be aware if it's a good environment, bad environment, need to move, respond to whatever it's sensing. So as soon as we start seeing the sensory organs increasing, then we start talking about the concentration of the nervous system. Therefore, we start talking about the formation of the brain in the front end of the body. Therefore, we start talking about the head. All right, so keep that in mind. It's extremely important, and it shows up in many groups of animals kind of independently from each other. We're talking about the divergent evolution. The head of, let's say, insect developed slightly different than my head, or head of the mammals. Why? Well, not why. Why they developed heads? Because they had the same, well, the ancestors of those groups had the same problem. We need to go into the environment, we have to sense it. When we are sensing it, we have to have some sort of brain. Brain or vision, for example, in insects is very different than the brain development and the vision as we have it in mammals or in vertebrates. Yet we have those particular heads, convergent evolution. So bilateral symmetrical will have anterior, the front end, the posterior, the back end, dorsal, upper part, ventral, lower surfaces, and then of course we'll have something which known as the, so here we have a dorsal, the basically the back side, and the ventral, the stomach side. For example, in our particular case, in the vertebrates, the spinal cord, it's even called spinal, the major nerve cord in our body, run along the body on the spinal, on the dorsal side. We have a dorsal hollow nerve cord. That's the vertebrates. Invertebrates, insects, for example, or crustaceans, they will have it on the ventral side. They have a ganglions running on the ventral side. Same kind of idea. We're talking about the convergent evolution. They had to have some sort of main highway made out of nerves, and it have to go throughout the body. Our ancestors as vertebrate ancestors ended up having it on the back, while the ancestors of the insects, well, arthropods, ended up being it on the front, on the ventral side. And we'll talk about lateral, we're talking about ventral, dorsal, yeah, lateral, lateral left, lateral right, all that particular part. All right, so tissues. Tissues are group of cells with a common function, which basically subdivide into animals into two major groups. We already talked about the sponges, not really true animals. Why? Because they don't have the true tissues. Therefore, it's called parazoa. Para, we were talking about paranthropuses. Remember, paranthropuses were near the humans. Anthropos means human. So, oh man. So in this particular case, they were developing parallel to us. In this particular case, parazoa near the animal. Zoa means animal, so near the animals. And eumetazoa or metazoa means true animals, means they have tissues. All right, so embryonic development will have two or three primary cell layers. We already talked about that. Remember the blastula, right? Later on, then there will be a layer which is moving in. That would be creating a second layer. And then there will be a third, some of the cells moving in between the outer and the inner layer, known as the layer in between layers. So diploblastic, only 2 layers, and triploblastic, 3 layers. And we'll discuss that. momentarily, and we'll do that. I'll show to you some of the examples in the lab number, well, next lab, number what, 11. So ectoderm. Ectos means outside. Ectoskeleton, we'll talk about that when we'll discuss the arthropods. So the outer layer, what the outer layer will form? Skin, external covering, and the nervous system. In most of the animals, if not all of the animals, the nervous system will be produced from the cells which are in the outer layer of the blastula, or I should say gastrula. Okay? Then we have the endoderm. This is the cells which are moving inside. They are responsible for the digestion. Therefore, pretty much all of the digestive organs, or most of the digestive organs, digestive tube, will be made out of that particular layer, endoderm. So we have the outer, we have the tube inside. So we're looking at the endoderm, ectoderm, now meso. What meso means? In between. In between of what? Ectoderm and the digestive tube. Anything in between. So if you will have an animal cross-secting, the outer layer is mostly made out of ectoderm. The digestive tube would be the endoderm. Everything in between, bones, muscles, and so on, some of the internal organs such as liver will be made mostly out of the mesoderm or developed from the cells of the mesoderm. Make sense? So everything in between the digestive tube and the external covering. Everything else, with some of the exceptions, of course, but everything else, specifically muscles, like for example, let's say jellyfish does not have much of the muscles. It has some, but not too many of those particular muscle cells. Why not? Because it's a diploblastic organism. It does not have a mesoderm. So most of the cells, muscle cells will not be developed. All right. So diploblastics will have ecto and endoderm. Triploblastics will have the ecto, endo, and mesoderm. So let's kind of take a look on the picture. It will be easier to see. So ectoderm, that's a cross section. So ectoderm outside, endoderm inside, lining up whatever the digestive cavity they have. And quite often in the middle, they will have a non-living layer, jelly-like material, which makes jellyfish jelly, literally. Now, in this case, in the triploblastic organism, we have the ectoderm and the endoderm, same thing. But now we have some cells moving in between of those, creating a third embryonic layer known as the mesoderm. All right. So triploblast will develop a third layer in between meso, like you probably heard about the Mesozoic era. It's between the Paleozoic and modern, ancient life, modern life, middle life, Mesozoic, mesoderm. Derm means layer, layer between, in between of what? Ecto and endoderm. Make sense? All right, so ectoderm, please remember that. There will be a question on the test. Ectoderm gives rise to the body covering, skin, and the nervous tissue. And skin is the largest organ in the human body. About what, 8 to 10 pounds of skin you have? Largest organ. So endoderm gives rise to the gut lining and most of the digestive organs. Not all, but most of the digestive organs. And then the mesoderm gives pretty much other body structures. We're talking about muscles, bones, blood, etc. Yes, blood is a tissue. It's a liquid tissue. We'll talk about that a little bit later. So triploblastic will be subdivided into a three. So we have animals subdivided into a diploblastic and triploblastic. Now triploblastic will be subdivided into a three different groups. Acoelomates. Now technically it's two groups: presence or absence of the body cavity. No body cavity, acoelomate. presence of the body cavity can be subdivided into two different groups known as the pseudocoelomates, false body cavity. And I'll discuss why it's considered to be a false and what's considered to be a true body cavity. And then this true body cavity will be subdivided again into the protostomes and deuterostomes. All right, so we are looking again at the cladogram, essentially. Number of layers, 1 or 2, 2 or 3, splitting, presence of mesoderm, no mesoderm. Here we're looking about the diploblastic, that's the derivative trait. Now we're looking at the branch with the presence of the mesoderm. Now presence of xylem, body cavity. No body cavity, okay, that's your acoelomates. Body cavity splits. Is it a true xylem? True body cavity, yes or no? If it's a true, then it splits again, developing the embryonic development, protostomes and the deuterostomes. Anyway, so let's take a look at the acoelomate. No body cavity. So let's take a look. We still have 3 embryonic layers. Everything is fine. We have ecto, meso, and endoderm. So ectoderm outside always will be outside. Endoderm inside, mesoderm in the middle. Now the mesoderm and endoderm do not form any cavity. There will be a, well, technically there will be a cavity which is filled up with the gelatin-like tissue. So there is no cavity per se, it's filled up. All right, no cavity means acoelomate. Example will be flatworm. Make sure you will know those particular examples. Many of the students on the short answers, I'm asking you, for example, give me the definition of biological species and provide me with examples. And some of the students give me a fairly nice definition, but lacking of the examples. No examples, I cannot give you the full credit. So flatworms. Flatworms, or something which known as platyhelminthes, that would be the example of the acoelomates. Very simple organisms. You literally can cut the liver fluke in half, and each one of those halves will regrow back into 2 separate worms. Now, pseudocoelomates. Now, what do we have? We have ectoderm, same as the before. We have mesoderm, we have the endoderm. But now we have that particular cavity. On one side, cavity is made by the mesoderm. On the inner side, it's made by the endoderm. So it's in between mesoderm and endoderm. And there will be some sort of organs, reproductive organs, for example, right here inside of that. Okay, considered to be a pseudo cavity. They have a cavity, but it's a false cavity. Why it's false? Because it's made out of mesoderm on one side, or bordering by mesoderm on one side and bordering by endoderm on another side. That's important. It will be on the test. Example, roundworms, nematodes. And then finally, a vertebrate, for example, or arthropods, the true coelomates. They have a true body cavity, which means what exactly? Look where the body cavity located at. We have the ectoderm and the mesoderm. The red is same. But now the body cavity is located within the mesoderm. Okay? The mesoderm completely lining up the body cavity on the inner and the outside of the cavity. Comparatively to the previous ones, here we have the outer body cavity is lined up with the mesoderm, inner with the endoderm. Here we're looking at the body cavity within the mesoderm, true body cavity. Okay? Any questions, any problems on that? We'll discuss that. I'll show you some more examples, but please make sure that you will understand that. If you don't, shoot me an email. I'll try to explain it to the best of my abilities. So pseudocoelomates were formerly classified as the separate group. We're keeping it as is, probably not a monophyletic group. Many of them are parasites, so we think that it's probably was simplification. At some point of time, the ancestors of the modern pseudocoelomates had a false cavity, but then they simplified the development. As a result, they ended up into a group known as a pseudocoelomates. We're not going that deep. We're not in zoology, even though zoology is my very, very favorite topic. That's my background in biology. I'm a zoologist. So in this particular case, we're not going into a comparative, too deep into a comparative embryology. Unfortunately, or fortunately, just don't have much time. So let's kind of take a look on all of those three things. So ectoderm stays exactly the same. Endoderm stays exactly the same. The thing is where the cavity located. Here we don't have the cavity per se. So no cavity, acoelomates, flatworms, moving on. Now in a false cavity, that particular kind of 10 color thing, the false cavity will be lined up, it's sandwiched between the mesoderm green and the endoderm red, okay? This is where the cavity located. This is where some of the internal organs will be located in. Now, for the true coelomates, the body cavities line up within the mesoderm or line up by the mesoderm. It's located within the mesoderm, all right? And make sure that you understand that acoelomates, flatworms, pseudocoelomates, roundworms, everything else you can think of, mollusks, the segmented worms, annelids, the vertebrates, the echinoderms, sea stars, etc., sea urchins, and the arthropods, insects, shrimp, crawfish, etc. All of those are true coelomates. So coelomates, no body cavity, pseudocoelomates sandwiched between the endoderm and mesoderm, and into a true coelomates, we're looking at the body cavity within the mesoderm. That is your answer for the question on the test number 3. Along those lines with the examples. So make sure you understand that. Make sure you will be able to provide the example of the phylum. All right, so now within the coelomates, we're looking at the protostomes and deuterostomes. I know it's a lot of theoretical material here. All right, so protostomes. We already discussed what exactly the protostomes and deuterostome means. Protos means first, stomos means mouth. The initial opening known as a blastopore, whenever the blastula start collapsing into the blastula cavity, into the cavity, will turn into the mouth. In deuterostomes, it will turn into anus, and the mouth will be secondary. So, but besides that, whenever the cell, the zygote, start dividing right from the, well, second division essentially, if we'll see something which is known as a spiral cleavage, if it's a spiral cleavage, you will be able to see that under the microscope, and the spiral cleavage is the characteristics of the protostomes. I'll show you the picture right there. Basically what we'll have, the cells will be kind of shifting at about 45 degrees, creating a spiral whenever they divide. Deuterostomes have a 90 degrees cleavage, so no problem. It's called the radial cleavage. So let's kind of take a look right there. So this is our 4-stage embryo, 4-cell stage. The very next stage, so 1 division, 2 division, on the 3rd division, you will see that the cells will not be exactly over the existing cells. They will shift a little bit, turn. As a result, from the top view, you will see that this is the original cells, the first four, and then we have a next layer. So spiraling up, so to speak. In the deuterostomes, we have exactly, so whenever you're looking from the top, you will see that the cells are cells located one under another. Next one, something which known as the predetermined or indeterminate division, and we'll talk about that, or a role of the cells. In the protostomes, each cell within a zygote is predetermined that it will turn. I'm simplified speaking. Simplifying speaking, let's say that particular cell is responsible for formation of the leg. I'm not, once again, just simplify it. It's not exactly that one for the formation of the leg, but let's say it is formation of the leg. And if we'll destroy it, that particular creature, let's say insect, grasshopper, will be born without the leg, because now you are destroying the predetermined. That particular cell is determined to become, to develop into the leg. You're destroying it at the early stages, no leg. And again, that's a very, very simplistic point of view, but this is what predetermined or indeterminate thing means. So now, on the radial cleavage, on the deuterostomes, that particular cell, let's say it's supposed to turn into a leg. If we destroy the nearby cells, will take over, and that particular creature for the deuterostomes will be born with the proper leg. Okay. So three major clades. We talk about the protostomes and deuterostomes. Within the protostomes there is a two major groups known as the Logotrophozoa and the Excidozoa. And we'll discuss it a little bit later. As a matter of fact, in one of the tables, in one of the labs, you will be creating the cladogram. Logotroph means the at the larvae development, they will have the round of tentacles, and in some cases the tentacles will stay as the adult creatures. So for example, mollusks. Excidozoa is the shading of the outer skeleton, or having the outer skeleton. So that's the roundworms as well as the arthropods. And the deuterostomes, that's basically other creatures. So let's kind of take a look on that, on the tree or cladogram of the animals. Choanoflagellates, right? Multicellularity. From the multicellularity, we start talking about true tissue. So periphery, the sponges moving out right here, they don't have a true tissues. Then the radial symmetry, Cnidarians and Ctenophora. And then we have a bilateral symmetry, all of them. And within the bilateral symmetry, we're talking about the protostomes or deuterostomes. And in the protostomes development, we have the exodus, the shading of the, the shading of the outer skeleton, and then having a lophotrough. As you can see, the platyhelminthes, mollusks, annelids, etc. For the exodus, we have two very large groups. Nematode, the roundworms, we talk about those ones in one of the labs, and then the arthropods, which is insects and crustaceans and spiders, so arachnids, all of that. Those guys we will kind of omit. I'll talk about Onychophora a little bit, but not much. But in the lab, we'll talk about the nematodes, we'll talk about the arthropods, we'll talk about the flatworms, we'll talk about the annelids, and we'll talk about the mollusks. So that basically what I will be looking later on that particular page. You will need to not copy per se, but based on that, you will need to work your way out for the filling out the table, the cladogram in one of the labs. So in the protostomes, part of the mesoderm separates. You don't really have to worry about that. It's kind of very, very theoretical, but very important. Remember when we start talking about the initial collapse, that particular opening known as the blastopore, and in the protostome, the blastopore will turn into a mouth, continue to develop, and will end up in the anus. So we will have the tube within the body. Now, in the deuterostome, that blastopore essentially will turn into anus, the back end of the digestive system, continue to go, and the mouth will form secondly. Therefore, the deuterostome. We belong, by the way, to the deuterostomes, humans, vertebrates. So here we have some of the examples of the protostomes, platyhelminthes, mollusks, annelids. About those ones we'll talk in the lab. We have the separate lab for which combined all of those. And the exodozoa, nematodes, and arthropods, we'll discuss that particular groups as well. So in the deuterostomes, echinoderms, starfish and their relatives, and the chordates. So phylum Porifera, the very first group of kinda animals. And we'll stop today on that. Well, we'll finish with the Porifera and we'll be done for today. So you will have some time to kind of study for the lab practical, right? So anyway, so phylum Porifera, choanocytes. Remember the colored cells? They have them. This is the first very simple group. So as a matter of fact, we're looking at this SpongeBob right here and the jellyfish. Jellyfish will be a next group we'll discuss, phylum Cnidarians, but that will be next time. So SpongeBob body will consist out of a sac which has a tiny little bitty opening. So the sponge sucking in the water, releasing it through the opening on top, known as the osculum, and we'll discuss that momentarily. And whatever is captured by, we're talking about small algae, bacteria, etc., the sponge will try to digest. Cells do not form through tissues. There is a several typical cell within the sponge body, for example, the choanoflagellates, but at some point of time, choanoflagellate will decide, I don't really want to stay here no more and wave that particular flagellum. So what I will do, I will lose it and I will become an amoeboid cell. Vice versa, amoeboid cell at some point of time will start growing the flagellum and will start beating, creating the flow of water. So again, asymmetrical, no definite symmetry based on whatever they in the environment they have. Body, well, acoelomate don't have the xylem. Body resemble the perforated sac, essentially. So if it's a sac, look, we have the walls, cross section. We have a walls, that particular inner part, inner cavity known as a spongiocell. It's not a body cavity. It's a cavity within the sponge, hence the spongiocell. Cell means, cell means cavity. So cavity within the sponge. So if we're looking at that, on top, they will have the main opening, large opening known as the osculum. So make sure that you understand that the water comes from all over, from the sides, and moved out. Some of the students think that the water comes in and then spreads out. No, 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 it's in the reverse. It will be, the sponge will gather the water from all over it to maximize the hunting ground, so to speak. And then it will send it out of the osculum. Lining the body cells, flagellated cells known as the choanocytes. So inside of that particular layer, there is a choanocytes, they beating the flagellum, creating the flow of water. So yeah, again, the flow of water goes through the pores, through the body wall, and through the spongiocell and out in the osculum. Composed of many elements, there is a mesophyll which creating the gelatinous material where the cells and the skeleton, internal, well, kind of skeleton parts of the sponge embedded in. And then there is the amoebocyte, so we have a choanocytes, and then we have amoebocytes. This is amoeba-looking cells which are going throughout the body of the sponge, capturing any kind of bacteria, algae, digesting, and then delivering the nutrients to the rest of the body. Okay, so motile cells, they capture, digest, and transport food. And then we have also something which is known as the spicules. Spicules are spikes, essentially embedded in a gelatinous material. That's the part of the skeleton. Most of the sponges are based, or classification of the sponges based on what type of the spicules they have, and we'll discuss that momentarily. So here, what we're looking at, sponge, right? And within the inner side, so here we're looking at the sponge, the body of the sponge, the cross section of the body of the sponge. And yes, it looks symmetrical. Technically, it's not. So that particular large cavity is a sponge cell, and this is the opening, which is known as the osculum. So the water comes from all over, will be sucked in. Why it's sucking in? Because those particular choanocytes beating the flagellus constantly. So they're moving water out. As a result, the water have to move in. Throughout the body of this sponge, there will be amoebocytes, which are capturing whatever is going through those particular channels. Therefore, they can capture the food and digest it. All right. So, and in this particular case, why it's not through tissues? Because again, at some point of time, choanocyte can change into the amoeboid, amoeboid can change into the choanocyte. Not as simple as that, but generally is. So here are some of the different cross-section of the sponge, the simplest one and the most complex ones. But the main idea is to increase the surface area. That's why you will have just the channel with the cavities and so on. So that one, of course, the interaction between the sponge, the inner part of the sponge and the water is drastically increased. So increasing the surface area, increasing the interaction. So this is the general cross-section of the sponge. And again, we're looking at the outer part, those particular spikes known as the spicules. They embedded throughout the sponge, creating the skeleton. So here, what we're looking at, the choanocyte, amoebocyte, and the spicules going through the sponge. All right. Now, sponges will have the skeleton, and based on that, there will be three major groups of sponges. Calcarea. Sponges have spicules made out of calcium carbonate. They're very heavy looking sponges. Usually that's not something which known as the bath sponge, which you can start using because it will be almost like a rock. And later on, you will see it under the microscope, the spicules of 3 rays. So everything is, a large lump of calcium carbonate. Okay, that's a calcera or calcium sponges. Then there is a demosponges. Spicules made out of silica. Remember, we talked about the silica SiO2, glass essentially. And also it will have demosponges, they will have the protein intertwined with those particular spicules. And this is where a more unnatural bath sponges coming from. And then finally, glass sponges or hexanellida, the rays are made out of silica or spicules, always a 6-rayed, and they pretty much lacking the protein component. As a result, it will be just really hard. They are really looking like a glass, very intricate. I'll show you. We'll cover them in the very first lab. You will see the glass sponge. So, and on that particular note, we'll stop right here. All right.